All right, so this is a propagation of action potentials. That's a nerve impulse. One of the reasons that you cannot propagate backwards is simply because of the previous patch happened to be in the refractory period, and therefore the voltage gated channels will not respond, and therefore it propagates only in one direction. Okay. All right. Now, what determines the speed? Okay. There's two factors that determine how fast that nerve impulse can go down the axon. One of them is the diameter of the axon. Okay, the wider the axon, the faster it is. So something that has a very thin axon, it would go more slowly. Okay, and so with a giant axon that's in the squid, that you can actually see it with a dissecting microscope. It's one of the biggest axons that we know about. Propagation is, they've measured it at 100 meters per second. Now obviously our neurons in our body, they're not this big, but nonetheless size, the diameter of the axon does determine how fast things go. And still, even for us with diameters that are much smaller than in the giant squid, the speed of propagation is real fast. Okay, it's almost instantaneous. Second factor is myelination. Okay, so if you remember the oligodendrocytes in the central nervous system, the brain, spinal cord, and Schwann cells in the peripheral nervous system, motor neurons, sensory neurons, create insulation, and this leads to the myelination is real important for how fast it can go. There are some neurons in the central nervous system in the brain that are not myelinated, okay, but they're very short distance. But most things are myelinated. And as a consequence, in this area where the myelin sheath happens to be, there are no voltage-gated channels there. The voltage-gated channels are focused right here in between myelin sheaths in that place that's called nodes of Ranvier. Okay, nodes of Ranvier, these are the gaps. And as a consequence, what happens here is a type of propagation is called saltatory conduction, because we'll have an action potential start here, and as long as the positive charge with the sodium inflow of the sodium comes in here, then it can, since down here is negative, this is positive, then that positive charge doesn't have to diffuse. Okay, the positive charge is on, of course, the sodium, but it's not Brownian motion. Okay, it's charge attraction. This is charge, this is negative down here, and this positive charge will jump real quickly. Okay, and lead to depolarization in this area, which then activates the voltage-gated channels in that particular node. And so you're getting this leap from one node of Ranvier to the next, and it's very, very rapid. What happens with Parkinson's disease is that it's an autoimmune disease that your body attacks these Schwann cells and destroys them. So you're losing the myelination, and as a consequence, a lot of times people eventually lose control of the skeletal muscle. Okay, because what happens is that the signal will dissipate. Okay, it won't make it to the end, the axon terminus, because of leakage of charge out through leak channels. All this myelination prevents a lot of that. So this affects the speed. All right, now looking at the synapse. This is where neuron makes contact with some other cell. A lot of times it's another, if it's in the central nervous system, it's an inner neuron, then it would be making contact with another neuron. But we also have nerve cells making synapses with skeletal muscle as well, okay, and cardiac muscle, and so forth. And so this is called a synapse, where we have this junction. And since we have two cells involved there, we can refer to one of them, the upstream one, as the presynaptic cell, and the one that is downstream of the synapse is the postsynaptic cell. And the synapses, there's actually two different types of synapses. One, which I haven't talked about, it's called electrical synapse. For the electrical synapse, we have the two cells, their cytoplasms are connected by gap junctions, okay? I mentioned gap junctions before when I talked about propagation of electrical signal through cardiac muscle, okay? So what this is that that synapse has no control over the electrical signal. So once we get depolarization running down to here, it just, those sodium ions can come in and go right into the next cell without hesitation through the gap junctions, because the cytoplasms are common, and it'll depolarize this second postsynaptic cell, and the voltage gated channels, they will respond. And so there's no hesitation. The electrical signal, the nerve impulse, just travels straight through without hesitation. So these types, we do have a few in our bodies, therefore reflexes, okay? So the nerve impulse just continues into the postsynaptic cell, and this is for usually very fast and stereotyped behaviors, okay? It's electrical synapses that control the backward flip of the tail of a lobster or a crayfish. Crayfish, they're crustaceans, and to escape, they have this reflex where the tail bends down and pulls it backwards real quickly. And that involves these types of synapses, okay? Electrical synapses. Other synapses are chemical synapses, where when the nerve impulse reaches the presynaptic cell, then it leads to the release of neurotransmitters from the presynaptic cell, and there's a narrow space. The plasma membranes are not touching each other. Plasma membranes are not touching each other. There's a fluid-filled space called synaptic cleft, and underneath the plasma membrane of the presynaptic cell, tethered to the cytoskeleton just under the plasma membrane, happens to be vesicles, like transport vesicles, but they contain neurotransmitters. So this has been a theme that we've seen many times now in this class, is that these vesicles, they contain, of course, neurotransmitters. And under the right signals, then these synaptic vesicles will fuse to the plasma membrane, releasing the neurotransmitter into the synaptic cleft. And that neurotransmitter has to fuse to the postsynaptic cell, where there happen to be receptors for those neurotransmitters, okay? And usually those receptors, they are chemically gated channels, okay? They're chemically gated channels. And binding of the neurotransmitter can do one of two things. It can either prevent those channels from opening, or stimulate the channels from stimulate them so that they open, okay? And what we have, and of course, the synapse, if it's with another neuron, it's the synapse is going to be on the cell body or on a dendrite, okay? Not on an axon. Okay, now, when this electrical signal in the presynaptic cell reaches the axon termini, we have here another type of voltage-gated channel that is not found elsewhere on the cell, only at the axon termini. And this happens to be for calcium. So it's a voltage-gated calcium channel. And so as the action potentials reach here, we get the influx, first of all, of sodium, okay, which depolarizes, which then stimulates these voltage-gated calcium channels to open, and calcium is more concentrated outside the cell. Okay, it's been pumped outside of the cell, it's not in the cytoplasm. Calcium floods in to the axon terminus, and just like with the cortical granules we talked about with fertilization of a sea urchin egg, calcium is always involved in membrane fusions. And so this will lead, the calcium influx will interact with these synaptic vesicles, lead to their fusion to the plasma membrane, releasing the neurotransmitter. Okay, so calcium is higher outside the cell. And cytoplasm is relatively, it has a very, very low concentration of calcium. So we also have a calcium ATPase that's in the plasma membrane. And so it will pump the calcium back out. So we have both calcium channels and calcium ATPases. Okay, and so this is what's happening at a chemical channel. Now, usually this duration of this neurotransmitter into the synaptic cleft is very short-lived. Okay, they're not there for very long. A lot of times, either they are taken up and degraded by the postsynaptic cell. Usually, however, they're taken up by endocytosis by the presynaptic cell, or they may have in the synaptic cleft here a enzyme that breaks the neurotransmitter down, and then the decay products of the neurotransmitter are retaken up and reformed by the presynaptic cell. They're usually recycled. So the one way in which antidepressants, usually antidepressants are aimed at channels, neurons that release the neurotransmitter serotonin. Serotonin seems to be very much connected to depression. And so a lot of these antidepressant drugs, they prevent the serotonin from being broken down so that they stay for a longer period here in the synaptic cleft so that they have their influence on the postsynaptic cell. And this is how those antidepressants are mostly working. Okay, now looking at the postsynaptic cell. So it has receptors for the neurotransmitters, and those receptors usually are connected, they are connected with a chemically gated channel, or they are on a chemically gated channel. Nonetheless, they're controlling the opening and closing of those gates. And what will happen, it leads to graded potential changes. Okay, all these chemically gated channels, they lead to graded potential changes. In other words, the magnitude of the depolarization or hyperpolarization, could go either way, depends on how many of those channels are open. And how many of those channels open depends on how much neurotransmitter was released. And how much neurotransmitter gets released really depends on how frequent the nerve impulses are arriving at the axon terminus in the presynaptic cell. The more rapid that they're arriving there, the more neurotransmitter that will be released. So this is how the strength of the stimulus is determined. Now these neurotransmitters, they may be excitatory or inhibitory, depending on what the neurotransmitter is and what type of channel on the postsynaptic cell that it binds to. And as I said before, the duration of that neurotransmitter being there is very short. It's broken down very, very quickly. And the breakdown products are recycled, usually into the presynaptic cell. Okay, so you tell me, you reason this out. The neurotransmitter is one that opens potassium channels on the postsynaptic cell. What would it lead to? What type of graded potential change? Because where's potassium? It's inside, right? It's in the cytoplasm of the postsynaptic cell. And so if we open up potassium channels, which direction is potassium going to go?
It's going to go out.
Out. It's going to go out. And so what's that going to do to membrane potential?
Go down.
It's going to become more negative. It's going to hyperpolarize. And so that's going to inhibit, it's pushing the membrane potential away from threshold, making it less likely that you're going to trigger any reaction in the postsynaptic cell. So this is an inhibitory. okay? So hyperpolarization, and so this particular type of graded potential change, these neurobiologists, they just love words and love vocabulary. So this type of hyperpolarization is called an inhibitory post-synaptic potential, or abbreviated IPSP, inhibitory post-synaptic. Now you probably saw that on the sample exam, okay? But it was on previous year's exam, but it won't be on this year's exam. It'll be on maybe the final, all right, that you'll see this. Okay, now what happens if we, so it's moving this away from the threshold, and so it's preventing the postsynaptic neuron from firing, okay? Now, what happens if we open, oh, let's jump back, no, let's go ahead with this. What happens if the neurotransmitter binds to sodium channels, chemically gated sodium channels, and opens them? Which direction in the postsynaptic cell will sodium go?
Should go in.
Goes in. And what does that do to the membrane potential?
It makes it go positive.
Okay, it depolarizes it, so it becomes less negative. Pushes the membrane potential toward threshold. Okay, because resting potential is what?
-70.
-70, and threshold is?
-50.
Okay, so this is pushing it toward, it's depolarizing, and it's a graded potential change. And what we have to do with that membrane, especially at the axon hillock, okay, the base of the axon, right next to the cell body, is that if the membrane potential at that part reaches threshold, -50, then it triggers the voltage gated channels to respond, and you get the propagation of action potentials.
I see.
So this is depolarization, and so this particular type of graded potential change is called an excitatory postsynaptic potential, or EPSP. This is moving to a threshold. Now, besides sodium and potassium channels, there are also chlorine channels, okay? Chemically gated chlorine channels. The concentration of chlorine is high outside the cell, low inside. High outside, low inside. Okay, so if we open up a chlorine channel, which direction would chlorine go?
It should go inside the membrane.
You'd go in, right? Because chlorine's higher outside. What would that do to the membrane potential in the postsynaptic cell?
It should make it less negative.
Say that again?
Make it less negative.
Okay, this has negative charge.
Oh,
Chlorine has
make
a
it
negative
more-
charge, and you're moving negative
Right.
into.
Make it more
it's-
negative in this case.
It,
Yeah.
it is also an inhibitory postsynaptic potential. Okay, it pushes it away from. Okay, now the number of inputs on a neuron can be pretty big. It can have hundreds of other neurons, some of which are inhibitory, some of which are excitatory. And so really what happens here, it's getting all this input, and this is an electron micrograph showing the cell body of a neuron with all these other neurons, axon termini, on its surface. So what does this poor neuron do? Okay, will it fire or will it not? Will it send out an impulse or will it not? It depends on the summation of all of these inputs and what happens down here at the axon hillock. Okay, some of these are inhibitory, some of them are excitatory, but how this neuron will respond is it will sum all of these inputs, and whatever, if it depolarizes at the axon hillock to -50, then we get action potentials.
How long does this usually take, the whole summation and stuff to like impulse?
What's that?
How long does it usually take for the neuron to like sum everything?
Oh, all this stuff is pretty fast. Yeah, all this stuff is really fast. Okay, so how do these things sum? Okay, now this here is making it a little bit, this figure is making it a little bit simple. Usually if we just have one neuron presynaptic that releases neurotransmitter, that's usually not enough to reach threshold. And so what we still have an excitatory postsynaptic potential here, but it decays real quickly. Now, we can have what is called temporal summation. What happens with temporal summation is we're talking about the same excitatory neuron, okay, the same excitatory neuron, but action potentials are coming more frequently. And so before the first release of neurotransmitter decays, another nerve impulse will reach the axon terminus. When they come close enough together, then it will release more neurotransmitter, and therefore those two nerve impulses that are arriving close in time, their effects sum together. And that can push it to threshold where now our voltage-gated channels will respond and we get the stereotype action potential. So that's called temporal summation. But there can also be spatial summation. In this case, we'd have two excitatory ones, one here, one there, different ones. It's still a matter of time too, but this, when we're talking about two different neurons that are on the cell body, giving their input, we still call it spatial. If they both happen to be firing at the same time, and their impulses, one is releasing neurotransmitter, we get a depolarization, graded depolarization, and second one is releasing a neurotransmitter as well. Those sum together, and so we get enough depolarization that reaches threshold, and we get our action potential. But another thing that can happen is also cancellation, because if we have an excitatory one and then an inhibitory one that are putting inputs into the postsynaptic cell, they can cancel each other. One of them is leading to a depolarization, graded depolarization, and an excitatory postsynaptic potential, and the other one is inhibitory, and they cancel each other out. And so we have all this type of inputs, and that lead to whatever happens to this neuron. And so you can see that the nervous system is very, very complex, the way it's put together, because we have billions of neurons in our brain. Okay, and the last slide on this, and I'm not going any further onto the nervous system, is different groups of neurotransmitters. Now, I don't expect you to know these, I just want you to know not each and every one, but these are groups. I just want you to know the groups. Now, there is one, I do want you to know this, acetylcholine. Okay, acetylcholine is a neurotransmitter that is found at neuromuscular junctions. Okay, neuromuscular junctions, which I'm going to be talking about shortly. Neuromuscular junctions. That's where the neuron has a synapse with muscle, okay, skeletal muscle, which I'll talk about next. That's very important there. Acetylcholine is also found at some places inside the brain as well, but it's the only neurotransmitter that controls your skeletal muscle. And then we have a group that are called biogenic amines, and so they're amino acid derivatives. Okay, they're not amino acids, but they're derived from them. That includes dopamine and serotonin. I already mentioned serotonin is real important with controlling depression. Dopamine is more excitatory. And, okay, then there are some amino acids. That the amino acid itself is a neurotransmitter. So aspartate, glutamate, glycine, these are three neurotransmitters that are also amino acids that you find in your proteins. Okay, so those amino acids serve both purposes. Here, GABA, this is gamma aminobutyric acid. That's also an amino acid, but this is one amino acid that you never find incorporated into proteins. Because I mentioned before that although proteins consist of 20 basic amino acids, we have a lot more. And some of these amino acids have nothing to do with protein structure, like this one right here. And you don't need to worry about whether they're excitatory or inhibitory. And then you have some that are neuropeptides. Okay, they're short chains of amino acids, and they are involved as neurotransmitters. Okay? Any questions? Now, I'm not going to go into anything else about how the nerves are arranged or parts of the brain or anything like that. We just don't have the time for that. Okay, yes?
Can you go over the last line, the difference between temporal and spatial again?
Yeah. Is that what you wanted to? Okay. All right. Okay, the summation with temporal summation. You're talking about the same input neuron. And so nerve impulses are arriving in short successions. Now, when the first nerve impulse arrives at the axon terminus, it leads to the release of some neurotransmitter, but then that neurotransmitter gets destroyed real quickly. Okay, and so if the next nerve impulse in that very same one comes real late, then you just get a little depolarization, and it's the neurotransmitter is destroyed. But if the nerve impulses are coming very rapidly, okay, frequent, and they're close enough together, the first one will release neurotransmitter, and before that decays, then more neurotransmitters released with the next nerve impulse. Okay, so that is temporal summation, talking about summing the signals from one and the same neuron. Okay, spatial summation, it still has a time component to it. That's what gets confusing here, is that, but we're dealing with two or more neurons that are providing input at the same time. So the first one is releasing neurotransmitter that leads to depolarization. The second one is also releasing neurotransmitter, which adds to it. They sum until you reach that. Now, obviously, if we had neurotransmitter or presynaptic neuron 1 arrive, and then we have a long period of time before the second one arrives, they're not going to sum. Okay, but here, when we're talking about spatial summation, we're dealing with two or more different inputs that their signals add together, if they arrive at the same time. Okay, does that make sense?
Yes.
Is that clear?
Yep.
Okay. Any other questions? Yes?
Can I see slide 35?
35? Yes?
For the EPSP and IPSP cancellation, so that would be depolarizing and polarizing?
Yeah, yeah. So you would have one, the excitatory one would be releasing a neurotransmitter that is opening sodium channels, and the other one, the inhibitory one, be releasing potassium channels. And so you'd be having a polarization or depolarization, hyperpolarization at the same time in cancer.
Okay.
Yeah. So that's summation for that. Cancellation. Okay, now one of the things that nerves control is muscles. And so I am going to, before I go to ecology, I do want to have a short say here about skeletal muscle. Now, this particular